Welcome to our videotape on bacteriology. I'm Paul Nicoletti. I'm a professor in the Department of Pathobiology at the College of Veterinary Medicine at the University of Florida. Bacteriology, the study of bacteria, is a specialized science, and of course it is impossible for us to cover this subject in the next hour or so. So we will limit our discussion to some of the genera that are important in veterinary medicine. But first, we should make a few comments about bacteria, in that, first of all, they are single-celled organisms that multiply by binary fission. We can see these bacteria under an ordinary light microscope, which is aided by staining techniques, usually by gram-positive or gram-negative organisms. This is part of our classification of the organisms. Bacteria in general come in three different shapes. They are the coccobacillus, as shown in this slide, and those can be either in chains or in small clusters. We also divide these into rods, elongated bacteria, which again can be stained, as sometimes those also produce spores. Thirdly, the spirochetes that are shown in this slide. Most bacteria are not pathogenic, so again we will limit our discussion to some of those which are. For diagnosing bacterial infections, we have a number of ways to do this, which we'll talk about in a moment. But first, let's look at the structure of a bacterium that sometimes may have a capsule. Not all bacteria produce capsules. Some bacteria have flagella, which are shown in the hair-like projections from this organism on the screen. Some bacteria also have what are called pili. This is the shorter structures there. They are sometimes surrounded by a capsule that has polysaccharide that we sometimes refer to as LPS, or lipopolysaccharides, from which some of the bacteria have their pathogenicity. Some bacteria, as I've mentioned, produce spores, as are shown in this slide. We can culture bacteria on artificial media. Most of the bacteria can be grown depending upon the nutrients which we supply to them. Blood agar, shown in our previous slide, is a frequent medium for culturing the bacteria, and it is our intent to isolate individual colonies of bacteria so that we can study these further, including examination for staining techniques, requirements for growth, and other factors that help us in the classification of the organisms. We can culture the bacteria in liquid medium or on what we refer to as solid medium. In the liquid medium, shown in the flask here, we can grow up the organisms and usually then subculture these off onto solid medium, as is shown in this slide, with the individual colonies so that we can study their characteristics. Not all bacteria are pathogenic, but those that are have a variety of different ways in order to produce the pathogenesis, such as the production of toxins. The gram-positive organisms may produce what are called exotoxins. These are protein substances that are liberated from the cells as part of the growth and the lysing of the cells. Some bacteria produce endotoxins. This is mostly associated with gram-negative organisms. The lipopolysaccharide, which I mentioned previously, that is released during the growth and the lysis of these bacteria, particularly in the gut or the enteron, is where much of this is located. Bacteria may produce enzymes such as those of the staphylococci, the coagulase, which reacts with the prothrombin to cause a necrotizing effects, thus producing suppuration or pus, if you will, and exudate. They also may produce hyaluronidase, an enzyme which breaks down the matrix between the cells and allows for the spread of the bacteria within the tissues that they have invaded. We can stain the organisms, as I have mentioned earlier. Here is an example of a gram-negative stain produced by a smear on a slide. For our diagnostic techniques, we may use a variety of methods. Most of these are directed toward examining whether antibodies have been formed as a result of the presence of the bacteria in the host. In this case, as shown on the slide, this is a gel precipitin test which utilizes the soluble antigens from a bacterium and allowing these to diffuse through an agar gel so that they may then react with antibodies that may be present in the serum. The reason for multiple lines in this slide is that there may be more than one antigen that diffuses at different rates, and again, where it meets antibodies is where this precipitin line is formed. We also may use tube agglutination tests or agglutination in a plate or a rapid system, which I'll show in a moment, but we usually mix these in a tube system, these meaning the test serum and the antigen being a suspension of the bacteria for which we wish to test for. If we have antibodies against the antigen or the bacteria, these will clump or agglutinate. They will then fall to the bottom, and we get a clearing in the supernatant of the tube that we are using. These are usually done in doubling dilutions, and on this basis we can develop a titer or the endpoint of the number of antibodies that may be present there, and these are very useful in finding if the animal has in fact antibodies against that respective antigen. We can also have a rapid plate system or a rapid agglutination, as is shown in this slide, and we usually use a stained antigen so that we can see that the reaction has in fact taken place. Another test is the complement fixation test that depends upon natural complement in the blood serum that we are using. We mix an antigen with our test serum, and if there is a reaction that takes place, this utilizes the complement. If a reaction has not taken place, then the complement is available for an indicator system using rabid red blood cells and anti-red blood cells antigen, and so if the reaction takes place, then we know the complement was available for this reaction, and it was a negative test in the first part of the system. We can also use an ELISA test, an enzyme-labeled immunosorbent assay, in which antigen is attached to surfaces. If there is specific antibody, these will combine, and in the presence of a indicator system, an enzyme, there will be a color change, and this is a way of detecting if antibodies are there. So now let's begin with specific genera. In this case, with no particular order, so we will begin with the Staphylococcus, or the genus Staphylococcus, and there are 4 organisms in that genus of veterinary importance. The more commonly isolated organism is Staphylococcus aureus, which causes a variety of suppurative processes in host, in one particular, in mastitis, for example, in dairy cattle, the most commonly isolated organism from mastitis. Staphylococcus intermedius, Staphylococcus epidermidis is, as the latter term would suggest, principally a infection of the skin. And then there's Staphylococcus hyacus, which produces a syndrome called greasy pig disease, which we'll show in a few moments. This is a slide of the udder of a cow showing the gangrenous nature of Staphylococcus infection. Usually these are more chronic infections, and we can detect these by culturing the milk. But it can be life-threatening and causing an acute septicemia in some animals. Again, more commonly, it is a chronic infection, and we may detect this on the basis of looking at milk quality, such as the test that is shown here, the California mastitis test, looking for nucleic acids in the milk that are present because of the Staphylococcus infection. We may examine the first milk coming from the udder, looking for changes in the milk as it is filtered through a small screen, giving some indication that mastitis may be there. For control of mastitis, there are a number of ways, but principally depend upon sanitation procedures, because this is an organism, Staphylococcus, that is found frequently in nature, and so we depend on trying to prevent the entry of the organisms into the cow's udder between our milking techniques. Finally, then, we may consider using vaccines, although vaccines against Staphylococcus infection have historically not been very efficacious. The Staphylococcus hyacus, which I mentioned earlier, may produce this infection of the skin of the young pigs. Again, the contamination would be from the environment and lesions caused on the skin of the pig by the what we call needle teeth of newborn pigs, and the damage to the udder of the sow, then causing infection in the baby pigs. The genus Streptococcus, shown in this slide, are organisms that grow in a chain, as is demonstrated here. These are gram-positive organisms that are shown in this slide, and there are a number of the species of the genus Streptococcus that do in fact cause disease. The first one of these, Streptococcus agalactia, as the name suggests, is associated with milk, again an organism that causes clinical mastitis, particularly in cattle. Then there are other organisms within the genus that may cause specific diseases, and 2 or so of those we will mention in a moment. We may classify some of the members of the genus Streptococcus by using agglutination tests, as is shown on this slide, and this causes a grouping of the organisms into what are known as Lancefield typing, and this helps us then in characterization of the specific species that may be a member of that group, and helping us identify the cause of whatever condition it may be. A specific disease caused by Streptococcus equi, called strangles in horses, a highly contagious disease, and causes a respiratory transmission and syndrome particularly in young horses, but may also affect other tissues in the body besides the respiratory tree. If so, this causes a condition much more serious that is sometimes referred to as bastard strangles, meaning that it is extensive in the body. The clinical signs of strangles in horses are depression, anorexia or the refusal to eat, fever, a nasal discharge, and there may be abscesses that are formed, frequently formed abscesses in the lymph nodes of the head. In this slide, we are showing an extension of the Streptococcus infection by showing abscessation in the lungs of this horse. Streptococcus suis produces a disease in pigs, as the name would suggest, and this is particularly serious in young pigs, resulting in a septicemia causing sudden death in the pigs. We may also have less serious effects of the infection by arthritis signs. In this slide on the left, you may be able to see abscessation in the joint that has been dissected or severed in this case. In the middle leg toward the bottom of the slide, you may be able to see an abscess in the leg of this pig caused by the Streptococcus suis organisms. Streptococci have an affinity for the heart valves, the valves of the heart, that may produce local lesions, granulomatous lesions, as is shown in this slide. Anthrax, the genus Bacillus, very much in the news as we speak. Bacillus anthracis being the cause. There is really only one organism within the genus that is of much major importance, and that is Bacillus anthracis. In animals, particularly in ruminant animals, may result in a rapid death and with an interference of the clotting mechanism of the blood, so that from the natural orifices, the nares, the anus, one may see frank blood oozing from these orifices and a suggestion that this may be anthrax. Anthrax occurs primarily in certain soil types in what we refer to as anthrax areas in the United States, and these are areas that support the spores that are formed from the bacteria in the presence of oxygen, and this is then where one may be or is more likely to see the disease in domesticated animals. The spleen may be enlarged because it has filtered the damaged red blood cells during the process of the disease. There is a rapid rigor mortis and a decomposition of the animal, so that in its virulent form, one will see a septicemia. This is a blood smear showing the bacteria, which are largely capsulated and largely do not form spores until they are exposed to oxygen. But for a diagnosis of anthrax, one could take a blood sample, smear it onto a glass slide, staining it, and then looking for the characteristic bacteria, the rod-shaped bacteria in this slide. In this slide, we're showing an animal that has died and rigor mortis of the animal. And in this case, you may be able to detect the swollen spleen in this sheep that has been severed, and the presence of the anthrax organisms having caused this enlarged spleen. We may also get hemorrhages on the heart muscle, what we call ecchymotic hemorrhages on the heart muscle, as is shown in this slide. In non-ruminant animals, monogastric animals such as dogs, horses, swine, the anthrax is usually not as virulent, and one may get swollen lymph nodes, hemorrhagic lymph nodes that are shown in this slide, but it may not be as lethal an infection as it is in the more susceptible ruminant animals. Anthrax can be controlled by the use of a vaccine, which is quite effective in its control. And this is used principally in those anthrax areas and also in other countries where anthrax is more commonly found than it is in the United States. In this slide, this is a smear of Corynebacterium organisms. This is a gram-positive genus and causes a variety of diseases. A number of the former members of this genus have been reclassified, and so there are two members of this genus which are currently of major importance in veterinary medicine. The first of these is an organism known as Corynebacterium renale, and from the name of the organism, one would suggest that this causes a problem in the urinary system. And the infection, an ascending infection, results in hemorrhagic mucosa in the bladder and also may ascend into the pelvis of the kidney, as is shown in this slide. So that there is hemorrhage, there is hemorrhage in the urine, and also hemorrhage from the urinary bladder, as is shown in this slide. This is a difficult disease to treat, and so animals that have Corynebacterium renale infection, and these are primarily females, are suggested that they probably should be culled. And another infection caused by a member of the genus Corynebacterium is Corynebacterium pseudotuberculosis that causes a disease called caseous lymphadenitis. This is a disease that is primarily of sheep or goats and causes a localized infection, usually of the external lymph nodes, where abscesses are formed in these lymph nodes and in the surrounding subcutaneous tissues. However, a more extensive infection could result in abscesses in the lungs, the liver, and other parts of the body. Here we see this goat with the parotid lymph nodes enlarged, abscessing, and this is then the most common way that we will see this disease in goats. In sheep, the prescapular lymph nodes are more commonly involved than they are in goats. In this slide, we show a lung of a sheep with the abscesses in the lungs from the Corynebacterium infection. The treatment of this disease is very difficult because of the formation of granulomatous tissues surrounding the abscess and the inability of antibiotics in sufficient concentrations to reach the organisms within the affected lymph node or other tissues. Rhodococcus equi, a former member of the genus Corynebacterium, causes a specific disease in horses from this gram-negative organism. It can be easily grown on blood agar, as is demonstrated in this slide, and causes a suppurative pneumonia and an enteritis and a fever and depression of this young horse, where the disease is most commonly seen. The organisms are soil-borne. In other words, they may be present outside of affected horses, so it is difficult to eliminate this organism from an environment. Here we see a young horse with Rhodococcus infection showing some depression, and in this horse, the nasal discharge, which is characteristic of Rhodococcus infections in horses. In this slide, we see a pneumonia, an extension of the Rhodococcus infection from the head, causing abscessation in the lungs. For the diagnosis of this, we can, of course, do a physical examination, and we can culture the organisms as I have previously described. We can do an ELISA test or an AGID test, looking for those horses that not only are infected, but may also be subsequent carriers of the organisms. In this case, this is an agar gel showing the precipitin lines that may be caused by the presence of antibodies in the serum of this test horse. For the genus Listeriosis, only one organism in the genus of much veterinary importance, and that's Listeria monocytogenes, so named because of its characteristic affecting the monocytes of the body and producing a syndrome known as paravascular cuffing. This is a flagellated organism, as is shown in this slide, and as I've just mentioned, produces a monocytosis of the small vessels of the brain. Characteristic, the disease is more commonly seen in sheep and is often associated with the feeding of spoiled silage. The mortality rate is very high in this disease, but the morbidity rate is usually very low. In other words, not very many of susceptible animals will in fact show clinical signs. There are 3 clinical syndromes of the infection, and it may cause an abortion. Incidentally, it may also cause abortions in women. Then there is the septicemic form causing the paravascular cuffing, which I've just described, and then also the encephalitic form, and those usually may also be found simultaneously. In the aborted fetus, there are characteristic changes in the liver that would suggest a Listeria infection. It is sometimes referred to as circling disease, and here is a sheep showing a head tilt, and if we were to observe this sheep with in nature, the sheep would probably be circling always in one direction. Erysipelas, or the genus Erysipelathrix, a disease called erysipelas, and this is primarily associated with swine. There are basically 2 forms of the disease: the acute form, in which there is rather rapid death, a fever, an inability to eat, and a purple coloring of the skin of the various areas of the body, and there may be abortions if the animal survives that long. In this slide, we show a dead pig with the froth from the nares, showing that there has been respiratory distress, and if looking at the postmortem or the necropsy of this swine, seeing the fluid hemorrhage in the bronchial tree and the respiratory distress that accompanies this. In the chronic form of erysipelas, we largely will see arthritis. We also may see diamond-shaped skin lesions and the so-called diamond skin disease, and we also may see lesions on the heart valves, as we previously described with the streptococci. Here in this slide, you may see the erosions of the articular surfaces of this joint that would be caused by the Erysipelathrix organisms. This is the so-called diamond skin. You can see the coloration has been changed of the capillaries, resulting in this rather characteristic diamond skin lesions. And then furthermore, the lesions that may occur on the heart valve in chronic infections of the erysipelas infection. For prevention of erysipelas in pigs, there are a number of good vaccines that may either be given intranasally or parentally to prevent the organisms are commonly found in the soil, so that it is impossible to eliminate the organisms by simply vaccinating or treating affected animals. The erysipelas organisms are quite sensitive to penicillin and some of the other broad-spectrum antibiotics. Tuberculosis, a historic disease and one for which we have had an eradication program for many decades. This is with the target organism being the Mycobacterium bovis. There are three major organisms of the genus Mycobacteria, and then a large number of other of the less important Mycobacteria. The first organism is the Mycobacterium tuberculosis, which is the human form of tuberculosis. Then there's the Mycobacterium bovis, and then the Mycobacterium avium, or the avium complex. The classification of members of the genus Mycobacteria is actually quite complicated. In any case, we should talk primarily about Mycobacterium bovis. Again, there has been an eradication program for many years for this disease based upon the elimination, the testing and elimination of those that are positive to tuberculin tests. The prevalence of the Mycobacteria in the Mycobacterium bovis in the United States is very, very low. These organisms are what are called acid-fast organisms. They have a special staining, and you can see the contrast in this slide of the red, which is the Mycobacterium that are stained, and then the blue background showing that, again, this change, the contrast in the staining of this suspension. Tubercles may be found in a variety of different soft tissues. In this case, you can see the extensive lesions in the lung of this animal. And again, these may be found in a variety of different areas. In this case, these are tubercles in the liver of poultry, the Mycobacterium avian organisms, which have largely been eliminated from the United States because of changes in the poultry industry. In the cow, the examination for tuberculosis begins with a tuberculin test injected into what is called the caudal fold. If the animal has a hypersensitivity to the Mycobacterium bovis infection, there will be a response. The site is examined at approximately a 72-hour interval. Because there is some cross-reaction between the Mycobacteria in the animal, then those that have a response like this receive what is known as a cervical test, as is shown on this slide, looking at the two spots there on the side of the neck, using two different antigens, and then the size of the reaction, then an indication of what in fact is the organism which it has the most sensitivity to. Animals that are considered positive are required to be slaughtered, and indeed most of the herds from which we now find the Mycobacterium bovis are in fact entirely slaughtered. We depend upon the detection of tuberculosis in our cattle population through the slaughter examination of slaughtered animals, and this slide simply shows the ratio of submitted specimens and those that turn out to, in fact, to be tubercular lesions. In other words, there are a number of possible causes of granulomatous lesions in the soft tissues of animals, and it is necessary to do histopathology, culturing, and other procedures for determining that this is in fact tuberculosis and not caused by parasites or neoplasms or some other cause. Another member of the genus Mycobacterium is the organism called Mycobacterium paratuberculosis, which causes a disease known as Johne's disease. This is a quite different disease than tuberculosis and affects principally the intestinal tract, more commonly the terminal part of the intestinal tract in cows and sometimes sheep and sometimes in other animals. So that it produces a granulomatous enteritis that results in a chronic diarrhea sometimes more acute, and this causes a weight loss in the animals, in other words, an unthriftiness, and occurs in cattle primarily that are over 2 years of age, even though the initial infection took place when they were very young. This is a slide showing the Mycobacterium paratuberculosis in a giant cell of the host, again showing that this is an acid-fast organism that may affect the various tissues. In this slide, you may see that the cow is somewhat emaciated, has a chronic diarrhea from the staining of the tail, the hairs of the tail in this case, and this is characteristic then of Johne's disease. Does not usually result in death, but certainly results in unthriftiness of the animal. A thickening of the bowel, as is shown in this slide, and again resulting in this chronic diarrhea and unthriftiness. We may test for Johne's disease in a variety of ways, including attempting to culture the organisms from the feces or from the tissues. We also have developed a number of serologic tests attempting to look for antibodies that may be specific against these organisms. The culturing is quite laborious and may not always be positive in the affected animal. The control measures for paratuberculosis are really quite extensive, and we cannot have time to examine each of these procedures. The important part is trying to prevent the young animal from becoming exposed to the organisms by mouth and then subsequently developing the disease some 2 or more years later. But it is a difficult disease to eliminate. It's growing in its incidence in the United States and will surely become a target of more control measures in the future. There has been a vaccine that has been developed, but is not very commonly used because it causes the development of a hypersensitivity that then may interfere with some of the diagnostic tests. Colibacillosis, or a disease syndrome that is caused by members of the genus, of the family, the Enterobacteriaceae, and in this case of the genus Escherichia. E. coli is very much in the news because of the importance in public health, which we will discuss in another tape. Colibacillosis then refers to the infection of these bacilli and as a result primarily of sanitation breakdowns. It results in a sometimes acute diarrhea, particularly in young animals, that may be cause mortality in these animals, particularly again in young pigs or in young cattle. It is a organism that has pili. Some of the species have pili and also flagella, and they attach to the villi of the gut, where they may cause damage to the epithelial cells, the loss of fluids, edema through the intestinal tract, then resulting, of course, in the diarrhea, as is shown in the watery feces in this slide. The signs then are maybe sudden death, dehydration, the failure to properly grow, and in the case of swine, there is a specific syndrome known as gut edema, where there is a swelling of the tissues of the stomach, as is shown in this case, and this frequently results in the death of the affected pig. In this case, this is the bone of a horse, and horses may get infections through the navel cord or the umbilicus, resulting in joint infections, as is shown in this slide, and in the young horse may be life-threatening or certainly causing arthritic problems. Treatment of the colibacillosis is primarily based upon the use of the replacement of the lost electrolytes and sometimes the use of antibiotics, but there is a tendency to develop a drug resistance by hosts that are affected by colibacillosis, and so it is discouraging to use antibiotics for the treatment of it. Vaccines have been developed and are primarily aimed toward giving the vaccine to the mother, to the dam, with the passage of antibodies through the colostral and other milk, hoping to protect then the newborn against colibacillosis. This is an example of a E. coli bacterin that may be given to the dam, the maternal host. Salmonellosis, also a member of the Salmonella, again a member of the Enterobacteriaceae, and there are a huge number of species, over 2,300 species in this genus that are largely typed on the basis of agglutination tests on the isolate using specific antibodies that have been developed in another host. These typing of the species of this genus is quite important in terms of the epidemiology of the disease, hoping that we can find the source through the identification of the specific organisms. This is a slide showing the agglutination or lack of it and the typing procedures that may be done. Salmonella affect a very large variety of hosts, some of which are shown in this slide. Almost all animals are susceptible to one or more of these salmonella, but characteristically the disease is one of the gut, but it may also cause septicemia, including the death of the animal. We may divide the salmonella into three major classes. Those that are primarily human pathogens, such as Salmonella typhi, the cause of typhoid fever. Those that are maybe adapted to a specific host, such as Salmonella cholerae suis and many others. And then those that are non-host adapted, such as Salmonella typhimurium and others, where they affect a variety of animal hosts, including humans. The characteristics of salmonellosis are, by and large, a rather high morbidity, in other words, a large number of the population to be affected, a low mortality, usually, depending again on so many factors, including the Salmonella serotype that may be present, but characteristically resulting in diarrhea, poor performance, and also may be a public health concern, which we will mention later. Here is the gut of an animal showing the sloughing of the membranes of the mucosa of the internal part of the gut. And here is the gut of a pig showing the hemorrhage and dilatation of the blood vessels surrounding the intestines from a salmonella infection. In some species, such as swine, salmonella have a tendency to cause ulcers that may in fact penetrate the gut, resulting in peritonitis. And in horses, there may be ulcers that form in the stomach as a result of salmonella infection. So salmonella can produce a whole variety of clinical syndromes in the various animals that it affects. As with the E. coli infections that we just mentioned, we discourage the use of antibiotics because of the possibility of causing resistant organisms to the antibiotics. On the other hand, if the disease is life-threatening, then certain antibiotics can be used. But it's primarily, meaning the treatment is primarily aimed toward the restoring of lost electrolytes, in other words, supportive treatment. As with E. coli, we can use vaccines largely in the same way that I mentioned about the colibacillosis, and that is trying to immunize the dam and the development then, or the passing of antibodies in the milk to protect the young animal. Pseudomonas, the genus Pseudomonas has only one organism of much veterinary importance, and in fact is not of high virulence, and this is Pseudomonas aeruginosa, that primarily causes external infections, such as that is shown in this slide of the ear infections in a dog that produces a rather obnoxious odor and may persist, and are very difficult sometimes to treat. The organisms are not very sensitive to some of the antibiotics, and so therefore have a tendency to become very chronic. The genus Campylobacter is primarily one of a reproductive syndrome, causes a disease which has been known as vibriosis and still often is, although technically should be referred to as campylobacteriosis. Primarily a disease of cattle that results in a clinical syndrome of abortion at various intervals in the gestation of the cow. It is a venereal disease, or maybe a venereal disease, in which the infection is transmitted by an infected bull that has bred an animal, a cow, and resulting then in a localized infection that may result in the death of the developing fetus. May be often in mid-pregnancy, but can occur at about any stage. In the aborted fetus, there are somewhat characteristic lesions in the liver of the aborted fetus, like white spots on the surface of the liver. Again, is a venereal disease and may be spread by the bull, where there are no clinical signs that develop. The disease is largely controlled through the use of vaccination. In other words, vaccinating the susceptible the susceptible animals prior to the breeding season, and hoping that this will then prevent infection. Artificial insemination can also be used since that eliminates the possibility of transmission by natural breeding. In sheep, there is an additional Campylobacter infection that is the result of oral exposure, and may also then result in abortion, showing characteristic lesions in this slide. There is also a syndrome in pigs called Campylobacter that may result in a diarrhea in the pigs, or in some instances no clinical signs, and this organism may also be of public health significance. Brucellosis, the genus Brucella. There are 6 members currently of the genus, one of which is of no veterinary importance at all. Brucella melitensis does not occur in the United States, but is the most important of the members of the genus Brucella around the world. Historically, this was the first organism that was described in Malta many years ago, and is of the major public health importance in many countries. In the United States, Brucella abortus affects cattle primarily, resulting in a clinical syndrome that primarily is one of abortion. Brucella suis, as its name would suggest, is a parasite of swine primarily, and has largely now in the United States become a disease only in feral pigs. Then there is a Brucella suis, a Brucella ovis organism that affects primarily male sheep, and then a Brucella canis, as the term would suggest, that affects dogs. Let's now show you a few slides of clinical brucellosis. In this case, this is an aborted fetus in cattle. Cows are curious creatures. they come to lick and smell the aborted fetus and then become exposed, so that the transmission is primarily one of ingestion. In sheep and goats, abortion also may occur, and again I would emphasize that this does not occur in the United States. Then in swine, Bacillus suis may cause a local abscessation, but characteristically also produces abortions in swine, and testicular lesions and epididymitis in the affected male swine. In dogs, in sheep, you may see in this slide the Brucella ovis, which, as I have suggested, produces testicular lesions, abscessation, and epididymitis affecting the fertility in sheep. Then in dogs, abortion in bitches and epididymitis, testicular lesions in the male dogs are characteristic of Brucella canis infections. Then in the United States, we also have a problem now with brucellosis in some wild animals, specifically elk and certain bison herds, and in elk, as is shown in this slide, abortion also resulting from the Brucella abortus infection. We can culture the fetus of an animal that is aborted looking for the Brucella organisms, and of course type those organisms to find out the specific etiology of this. More commonly for a diagnosis, we depend upon antibodies in the serum and can use a rapid or more extended tests. In this slide, we're showing the card test, which is a very rapid test and depends again upon the antibodies that may be present in the serum. In this case, this is a test for Brucella canis, a rapid test that simulates that can be used in other species. There is a ring test that is performed on all of the dairy herds in the United States, and this then relies upon the presence of antibodies in the milk of an infected animal, and if the test is positive, it is performed on the bulk samples or the milk of the entire herd, and then individual blood tests would be performed on the herd in which there was a positive ring test. There has been an eradication program in the United States for many, many years for Brucella abortus and Brucella suis, and it depends upon the slaughter of affected cattle. The incidence of brucellosis now among cattle in the United States is very low, as it is with swine, with the exception, as I previously mentioned, of feral swine. The control of brucellosis can also be aided by the use of vaccine. Historically, in cattle, the vaccine was known as strain 19. That has largely given way to a newer developed vaccine called RB51, which in fact does not cause the development of post-vaccinal antibodies, long a historic feature of strain 19, and one for which there were a lot of diagnostic problems in differentiation of those antibodies. In the genus Actinobacillus, one organism of importance, and that causes a localized infection of soft tissues where there may be abscessation, as is shown in this slide. A difficult disease to treat, but not one of life-threatening consequences. Then in swine, there is an organism called Actinobacillus pleuropneumoniae, which, as the name suggests, does cause pneumonic symptoms, particularly in young pigs again, and causing a syndrome that is shown on the slide of respiratory distress, and may result in the death of these pigs, as for reasons as you can see in this slide, and that is consolidation and other lesions in the lungs of the affected pigs. Bordetella, the genus Bordetella bronchoseptica, produces two clinical syndromes in two different animal species, in dogs producing a syndrome known as infectious tracheal bronchitis, the so-called kennel cough, and as the name suggests, causing a respiratory syndrome in dogs. And in swine, a syndrome known as atrophic rhinitis, and we'll show in a slide in a moment why it is named this. These are gram-negative organisms. There seems to be no relationship to the disease in swine and the disease in dogs. Nevertheless, it is the same organism, and these adhere to the cilia of the respiratory tract, as is shown in this slide, and the control is aimed toward preventing this adherence through the use of intranasal vaccines. This is a section of the nose of a swine that's been affected by the Bordetella organisms, and you may be able to see the distortion of the nasal septum in this case because of the damage, hence the term atrophic rhinitis. Again, vaccination can be done intranasally in the dogs and in pigs, and these are quite effective in controlling the Bordetella infections. The genus Haemophilus, primarily associated with a species, Haemophilus somnus, that a gram-negative organism that produces a syndrome, several different syndromes, primarily in cattle. One principally of a respiratory syndrome, in which is sometimes part of the respiratory disease complex, or the so-called shipping fever complex, in which there are lesions in the pulmonary system that results in pneumonias, sometimes the death of the animals. Then there's the septicemic form, which largely produces encephalitic signs, and then the reproductive form that may result in abortions. In this slide, we see pathology of the lungs that is caused by the Haemophilus organisms, and frequently assisted by members of other bacterial genera, especially Pasteurella, and also some of the viruses that may also be present in this syndrome. In the encephalitic form, as is suggested, the brain may be affected. There are abscesses that are developed in the brain, and this is a very serious part of the syndrome that often results in the death of the animal. In the reproductive form, abortions again may occur at almost any stage of the gestation and result then in reproductive failure for a period of time in that particular herd. Contagious equine metritis, or the Taylorella, in the genus Taylorella, and one member of this Equigenitalius. We do not have this disease in the United States, but it is of importance because of the international movement of horses. May, in its clinical form, produce a copious amount of exudate from the cervicitis and also from the uterine infections. Does not clinically affect the stallion, but the stallion can be carriers of the organisms. Again, it is of importance primarily in the regulatory area in the international commerce of horses. The genus Moraxella is one member. There is one member of this genus, Moraxella bovis, that causes what is usually referred to as pink eye or infectious keratoconjunctivitis, and occurs primarily in animals of a lighter skin, in white-skinned other Hereford cattle, and also as a result of infections that occur in the summertime in animals that are in sunlight, also those that may be affected by face flies or other vectors of these organisms that may transmit the disease from one animal to others. There are vaccines that may be used to control this. Here we see the clouded cornea of an animal, and sometimes this may even in fact result in blindness of animals that may be affected. Many of these will recover if taken out of conditions that have aggravated the infection. Again, vaccines can be used in the control. In the genus Pasteurella, there are two organisms that we should consider: Pasteurella hemolytica and Pasteurella multocida. I should at this point out that there has been a recent change of the generic name of the Pasteurella hemolytica, and the new generic name is Mannheimia, but we will refer to these two organisms today in the genus Pasteurella. Primarily a syndrome in the bovine respiratory complex that I mentioned when I mentioned the genus Haemophilus, and characteristically resulting following the movement of animals, the stress of animals that may be going into feedlots, etc. Characteristically then a fever, a coughing, and the respiratory complex, which, as I suggested earlier, may be the result of multiple causations. Here we can see an animal that has difficulty breathing, its head is extended, and then in the lungs we may see consolidation, edema, in other words, a difficulty of respiration, and resulting sometimes in the death of the animal. Again, these can be controlled partially through the use of vaccines, although historically these vaccines have not been very effective in the control of this. In the genus Clostridium, there are a number of organisms of importance in veterinary medicine. Some of these are listed on the slide, but prior to these we should mention two organisms, Clostridium tetani, that causes tetanus in animals, particularly in horses, and Clostridium botulinum, which causes a syndrome in a number of animal species. Here we see the Clostridia, which are gram-positive rods. These are Clostridium tetani that are growing in the tissues. As I mentioned previously, horses are particularly susceptible to tetanus, but other animals may also be. Tetanus usually follows some insult, some surgery, or a puncture wound, and is very frequently fatal to the affected animal. Here we see tetanus in a calf with its head pulled back in a characteristic muscular contraction that is characteristic of tetanus infection. Clostridium botulinum, an interesting organism historically, and as you surely know, has been an organism that has in the past been associated in humans with improperly canned foods. We don't see botulinum infections in humans only rarely anymore, but we do see botulinum sometimes in poultry, where it is referred to as limberneck. This is especially important in migratory waterfowl, where there may be huge die-offs of these from contaminated lakes and areas like this. The other Clostridia are more invasive in their tissues, and the first of these that we should consider is Clostridium shovii that produces a syndrome called blackleg. This is primarily a disease, almost always occurs in animals less than 6 months of age, and frequently a very rapid death. There is spore formation, which contaminates then the soil, And so the areas where blackleg occurs, as with other clostridial diseases, are considered to be contaminated for a long time. In this particular case, this is, as all the Clostridia are, is an anaerobic rod, and it localizes in muscles where it may produce gas formation and also hemorrhage, as is shown in the musculature of this calf. Another disease which is similar is malignant edema caused by the Clostridium septicum. This one often follows some insult and is characteristic by edema and of the stomach, as is shown in this slide. So Clostridium septicum is very rarely reported now in the United States, and there are good vaccines for both of these conditions, that is blackleg and malignant edema. Clostridium hemolyticum produces a syndrome that's sometimes referred to as red water disease, and this is simply because of the hemoglobinuria that the organisms may produce. There is a very high mortality rate. This meaning the disease, primarily occurs in certain areas, particularly areas where snails and liver flukes may be found. The disease may produce a characteristic infarct in the liver, as is shown in this slide. It is a disease that is not easily controlled through the use of vaccines, but vaccines are used in certain areas where this is most likely to occur. Finally, there is the Clostridium perfringens organism that produces a syndrome in a variety of different animal species, particularly in cattle, but also in sheep, also in swine, known as enterotoxemia. From the very name of this, it would suggest that the toxins are released in the enteron or in the gut. There are a number of different syndromes that are classified from A through D and usually result in rapid death of the animals and soft kidneys and a hemorrhagic enteritis. The control of this is usually through vaccines that are administered prior to the time that these animals are put into feedlot conditions. So the change in diet is one of the predisposing factors for the development of enterotoxemia in susceptible animals. Here you can see in the gut of this pig hemorrhage in parts of the gut, a characteristic of the disease. Here you may see in this slide the changes in the kidneys, so that it is sometimes referred to as pulpy kidney disease, again because of the changes that occur. The genus Actinomyces, one species of particular importance, Actinomyces bovis, and this produces a localized abscessation in the hard tissues or bony tissues, and these are deep abscesses, very difficult to treat, and follow wounds usually in the mouth from plant awns or other insults to the mucous membranes of the oropharynx, resulting then in the penetration of the organisms and abscessations that may be formed. Here you may see in this cow a swollen area on the side of the mandible, showing the area where the pus has come forward, and also they heal over, but then the abscess persists. Actinomyces pyogenes is another organism that was at one time in the genus Crenibacterium and is frequently isolated in what we call suppurative processes, that is those in which pus formation occurs following various insults, such as with hardware disease in cattle, where there may be penetration of objects into the soft tissues, in this case into the heart area, and large amounts of pus that is formed as a result of these actinobacillus organisms. This is a slide showing the organisms here that where the pus formation that may be quite copious in these areas. Actinomyces viscosus occurs primarily in dogs, particularly those that are outdoor dogs, that are hunting dogs, and usually follows an injury in the skin, and usually is a localized abscess, but sometimes may result in systemic infections. There are two genera that result, the Fusobacterium and the Bacteroides, that may cause what is referred to as foot rot infections. These are primarily in the cloven-footed animals, where there is a penetration of the organisms through the hoof and causing an abscessation of that area, resulting often in lameness that may be permanent in these animals. Frequently occurs in conditions that are less than sanitary, and may also in some animals, particularly in feedlot animals, result in liver abscesses, as is shown in this slide. There are vaccines that have been developed to control these conditions in sheep and in cattle. Finally, the genus Leptospirosis or Leptospira. There are a number of the organisms that, number of species within this genus that may affect a variety of different animal hosts. Leptospira are frequently invade the tubules of the of the kidneys, and you can see in this slide the contrast staining of the organisms, the black organisms in the renal tubules of this rat, where the rat is a very important host of the Leptospira, and may also affect public health, which we'll refer to later. In any case, here are a number of the Leptospira species and some of the hosts that they may affect. And while there is cross-species infections, nevertheless, many of the Leptospira do have preferential hosts, as is shown in this slide. Characteristic leptospirosis, it causes abortions, and these may occur at any time. They also may cause mastitis in cattle, and this abortions, of course, would result in lower milk production, as with the mastitis, of course, and there may be some infertility associated with this. There is usually not many clinical signs other than what we have suggested. Then in dogs, there may be an ecteric form of this causing depression and affecting the liver and the kidneys, resulting in icterus and sometimes the death of these. For diagnostic procedures, there are tests that may be performed looking again for antibodies in the serum of affected animals. This is called the microscopic agglutination test, which is performed on live antigen in some of the diagnostic laboratories. The control of leptospirosis in farm animals is primarily through routine vaccinations. This is particularly true in dairy cattle, where leptospirosis may be a real problem in causing mastitis and in abortions in the cattle. So vaccination becomes necessary in these conditions. The Leptospira may survive for some periods of time in water, in other words, moist conditions. Also, as I suggested earlier, the rat is important in leptospirosis in some instances, and so rodent control would be part of this. Finally, then, the genus Borrelia cause the Borrelia burgdorferi, the spirochete being the cause of Lyme disease. Lyme disease is a result of a spirochete infection and was named for Lyme, Connecticut, where the disease was first described. It may affect a variety of different animals, but is of particular importance in humans, which we will discuss in another tape. The vector of the disease is the Ixodes tick that is found in certain areas of the United States more than others, and over 90% or so of the human cases of Lyme disease occur largely in the northeast part of the United States. And again, this is associated with the prevalence of the Ixodes scapularis, the vector. Also in the upper Midwest, in Minnesota, Wisconsin, and then some in the northwest part of the United States. The principal reservoir, animal reservoir of the Borrelia is the white-footed mouse, shown in this slide, and some of the control measures are directed toward that particular animal species. Well, in the dogs, there may be arthritis, there may be fever, enlargement of the lymph nodes, pain, but it is not an easy disease to diagnose in dogs because of the limitations of some of the diagnostic tests. These are rather difficult to perform to be completely accurate about the diagnosis of this, and we've listed some of the diagnostic tests that can be performed here on this slide. The disease may affect cattle, although this seems to be fairly rare, resulting in arthritis, swollen joints, as is shown in this slide. There are vaccines that are available for use in dogs, but primarily are recommended for areas in which the disease is much more prevalent than others. Well, this concludes the discussion of some of the bacterial genera that are important in veterinary medicine. We simply do not have time to cover not only all of the genera, but also some of the other organisms that are classified by either the chlamydia, the rickettsia, the bartonella, the anaplasma, and the mycoplasmas that may also be important in veterinary medicine. I would encourage all of you to review the diseases that may be caused by not only some of the genera which we have not covered, but particularly those diseases that are caused by the other organisms that I have just mentioned. Good luck to you. It's been a pleasure to present this videotape.